So I'm going to give you a pretty quick review of meiosis, and you'll want to spend some time looking at this and reading about this more closely because it's important for the understanding of inheritance, since meiosis is the kind of cell division that produces haploid cells, and in animals directly it produces gametes. In fungus and plants, it produces spores, which then go on to produce gametes. But it's an important first part of sexual reproduction for all of the eukaryotes. And the second part of sexual reproduction, after meiosis forms gametes, then the second part is fertilization. But meiosis is important, and it is the first step. Meiosis consists of two divisions, back to back. The first division is called meiosis 1, and it consists of prophase 1, metaphase 1, anaphase 1, telophase 1. And it is a reductional division because the product cells at the end of meiosis 1 are haploid. So if you start with a diploid cell, by the end of meiosis 1, you have two cells, and each of those cells is haploid. And you can see that in the middle of the two boxes on the screen. So you start with the diploid cell. 2N is just a notation that means diploid, so two sets of chromosomes. Each set is color-coded, so the red large and the red medium chromosome makes up a set, and then the blue large and the blue medium is the other set. Sometimes in these drawings, colors are used to emphasize certain things. In this image, it's emphasizing the sets. So after meiosis 1 is completed, you have two daughter cells, and these are now haploid, one copy of each type of chromosome, a large and a medium chromosome in each cell. And you'll notice that the cells start off with chromosomes in the dyad form because DNA replication precedes meiosis 1. So the chromosomes start off in the dyad form. By the end of meiosis 1, they are still in the dyad form, but the cells have become haploid. And then each of those cells enters into meiosis 2. Meiosis 2 is the equational division because each of those cells starts with two chromosomes, haploid, but two chromosomes, one large and one medium. And then by the end of meiosis 2, each of those product cells also has two chromosomes, one large and one medium. However, the form of the chromosomes changes. At the beginning of meiosis 2, the chromosomes are in the dyad form, and each of the product cells at the end of meiosis 2 has monad form chromosomes. One thing that's also not being shown here is the crossing over, which you may recall happens at the beginning of meiosis 1, specifically prophase 1. So the way that the chromosomes line up after the crossing over happens, the way that the chromosomes line up is different in meiosis 1 versus meiosis 2, specifically metaphase 1 and metaphase 2. In meiosis 1, the chromosomes are in their homologous pairs, and they stay in their homologous pairs. They come together in a process called synapsis in prophase, and then crossing over is the breakage and exchange of equal corresponding segments on non-sister chromatids. And then the homologous pairs line up at the metaphase 1 plate. And you can see that in metaphase 1, how the chromosomes are lined up. In this cell, there's only 4 chromosomes, but it could be more than that. But here you have 2 pairs of chromosomes lined up at the metaphase plate. And anaphase 1 separates the homologs away from each other. So one large blue and one large red get separated from each other. One medium blue and one medium red get separated from each other. And then telophase 1, each of those groups reforms into a new nucleus, and cytokinesis separates the cytoplasm. In prophase 2, starting from that same point, prophase 2 involves the alignment of now just the dyad chromosomes in each cell. the dyads align individually at the metaphase plate. There is no significance to the fact that in this artist's drawing, they have the metaphase plate of meiosis 1 going vertically and the metaphase plate in meiosis 2 going horizontally. That's not meaningful. It's just the choice the artist made. What's important is how the chromosomes are lined up at the metaphase plate. In metaphase 1, they're still in their homologous pairs, and you can see that the metaphase plate splits down the middle between the homologous pairs. Anaphase 1 does not pull apart any of the centromeres. But in metaphase 2, the chromosomes are dyads, and they align so that the metaphase plate does split through the center of their centromere. And so when anaphase 2 pulls apart the chromosomes, it's pulling apart the sister chromatids away from each other. Meiosis 2 is a lot more like mitosis, in that you start with a cell that has, say, 2 chromosomes. You end with 2 cells that have 2 chromosomes and each. And so it's not that different than the way that the chromosomes are separated during mitosis. What we really want to do, though, is make sure we're very clear when we're comparing mitosis and meiosis what the purposes are. I mentioned the purpose of mitosis and cell cycle for growth of the organism to make more cells of the exact same kinds, for repair and growth. Meiosis, the purpose of meiosis is to produce the haploid cells, which eventually will be the gametes, which is the first step in sexual reproduction. And so that's quite a different purpose. The events in meiosis that do not occur in mitosis, those would be synapsis and crossing over in prophase 1, alignment of tetrads in metaphase 1, and separation of homologous chromosomes in anaphase 1. So pretty much everything in meiosis 1 is something that does not happen in mitosis. Usually crossing over, which happens in prophase 1, is cited as the thing that creates genetic variation among the gametes. And that's true. That's part of it anyway. Genetic variation at its very basic level starts with DNA mutations. So somehow DNA mutations happen. Some of them are naturally occurring. We call them spontaneous mutations. Some of them are induced by chemicals or radiation. We call those induced mutations. But whatever causes the mutation, a mutation in a gene creates a new allele of that gene. Now, once there are different alleles of genes, the alleles of different genes are mixed together into a lot of different combinations by these processes I list here. Prophase 1 crossing over and independent assortment in metaphase 1. So in this figure, you see what happens when genes are on the same chromosome. So gene A and gene B are on the same chromosome in this example. When there's crossing over, you get new combinations of those alleles. So for example, the big A and the big B was a combination in the starting cell. The little a and the little b was also a combination in the starting cell. But when crossing over happens, some of the newly formed chromatids now have new combinations of those alleles, because now you have a chromatid that has a little a with a big B all together now on the same chromatid, and also a big A connected to a little b on one chromatid. And so those are new combinations of alleles that weren't connected together on the same chromosome or on the same chromatid previously. So in the end, you get 4 gamete types. One of them has big A with big B, one of them has little a with big B, one of them has big A with little b, and one of them has little a with little b. And so crossing over gives you a new combinations of alleles of genes that are on the same chromosome. So there's a gene A, which has 2 alleles, and a gene B, which has two alleles. So when I say two genes on the same chromosome, I mean gene A and gene B are two different genes on the same chromosome and different allele combinations of those. Independent assortment deals with the combinations of all the chromosomes that are in the cell. So if we color coded all the chromosomes that a long time ago came from your mother in the egg and a long time ago came from your father in the sperm, they're all your chromosomes now. But if we color coded them to kind of track which ones were sort of originally from your mother and which ones were originally from your father, and we look at those in your cells, and your cell is going through meiosis, then what you'll see is the way that the chromosomes line up at meiosis has a lot of possibilities. And we could get, this is just 3 pairs of chromosomes, what they're calling chromosome 1, chromosome 2, and chromosome 3, and 2 copies of each. And so what you can see is they're putting the M, that's the chromosome from the maternal side, your mother's, but it's also yours. And the P is the chromosome that came from your paternal side, from your father, but also yours. So they're color coding those. All of these chromosomes are yours, but when your cells go through meiosis to create gametes, the gametes contain mixtures of red chromosomes and blue chromosomes. In this case, the red is indicating chromosomes that were originally from your mom and blue chromosomes that were originally from your dad. And what you end up with is different combinations possible of these chromosomes. In this picture, there's only 3 pairs of chromosomes, but humans have 23 pairs. So I think you can see how many different possible combinations could be possible in terms of this process. This is called independent assortment. So independent assortment produces by itself, because humans have 23 pairs, 8.4 million chromosome combinations that are possible in any gamete. Now remember that 2 gametes have to fuse together to create a new zygote. So the fusion of 2 gametes randomly could be any of those 8.4 million fusing with any other 8.4 of the 8.4 million. So you end up with a possibility of 70 trillion different zygote combinations, diploid combinations of chromosomes. It's very hard to quantitate crossing over, but crossing over happens more than once with the long chromosomes and at least once with the short chromosomes. And so when we start with a number of 70 trillion and we multiply in the amount of variation that's added by crossing over, the number becomes astronomical. And what this means is that no one else has the exact combination of alleles that you do, except unless you have an identical twin.